Good day. I'm Max Hegblom, Editor-in-Chief of FEMS Microbiology Ecology, and it is my pleasure to welcome you to this webinar on insect microbiome interactions. Our continuing webinar series enables us to highlight different topics of microbial ecology to a broad audience, you. The mission of FEMS, the Federation of European Microbiological Societies, is connecting, promoting, and supporting microbiology through knowledge exchange and community building for a globally sustainable future. Those of you who have turned into our previous webinars know that FEMS invests in science, using the income from our journals to fund charitable activities and support our community. Learned societies and their journals provide grants to scientists, organize and support conferences and summer schools, and sponsor a range of events, such as a webinar series like this one. These provide a forum for the presentation and discussion of key research, enabling the flow of ideas, connecting a worldwide audience. As an author, when you publish in FEMS journals, and as a reader, when you use these publications, you directly support the various activities in which FEMS invests back in science. You probably also know that from January this year, our journals have transitioned to be fully open access. This means that all the archives are free to read regardless of your institutional subscriptions. Please note that to ensure that article processing charges do not become a barrier for authors, we have a number of discounts and waivers available. Please see the journal website for more information on this. Today, our three speakers will take us on an exploration of the buzzing world of insect microbiome interactions. Our speakers will explain how microbial communities influence the lives of various insects, from bees to bark beetles. First, Danielle Rutkowski from the Department of Entomology and Nematology, University of California, Davis, will tell us about the beneficial fungal associations with bees, revealing their impact on bee behavior, health, and ecosystems role. Insecticides, of course, but also herbicides can have a major impact on insects. Our second speaker, Suni Matthew from the Department of Biology, University of Turku in Finland, has explored the effects of glyphosate on bumblebee gut microbiota, highlighting potential risks to bee health and biodiversity. And lastly, Teresa Veselska from the Institute of Microbiology of the Czech Academy of Sciences will reveal some of the dynamic changes in the gut microbiome of European spruce bark beetles across different life stages and seasons. So we hope that you will enjoy this webinar, exploring the complex interactions between insects and their microbial partners. After the three talks, we will open the sessions for questions and discussion. So please submit your questions via the Q&A link, and we will get back to these at the very end of the session. And before we start, I also wish to thank the staff of FEMS and Oxford University Press for all their work that is happening behind the scenes in making these webinars happen. So welcome, everyone, and it's my pleasure to hand over the podium or the microphone to Danielle, and we'll get into bees. So thank you.
Great. Thank you so much, Max, for the introduction. Let me share my screen here. Okay. So thank you all for coming here today. I'm Danielle, and today I'm really excited to be talking to you all about our recent review paper, Bees Just Want to Have Fungi, a review of bee associations with non-pathogenic fungi. So I probably don't have to explain to you all the importance of bees, both in an agricultural sense as well as for ecosystem services. So bees contribute to global crop pollination services that are worth billions of dollars annually around the globe. And in addition to their agricultural worth, bees also help pollinate the roughly 87% of flowering plant species that rely on animal pollination to some degree for their success. So the importance of these pollinators, both agriculturally and environmentally, is especially alarming when we consider that many of these species are actually in decline. So this data is from 2020, and it was particularly looking at bumblebee declines around the globe. And the authors found that depending on region, anywhere between 12 and 45% of bumblebee species are threatened in some way, according to IUCN. And of course, this data isn't representative of all bees. It's just looking at bumblebees, and you can see that it's incomplete and that we're missing certain regions of the world. But these declines are alarming, and they really point out the need for understanding the biology of these pollinators more completely so that we can create conservation strategies to protect them during these alarming declining periods. And one particular region or field of bee health that has received a lot of attention lately has been bee microbial ecology, or the interaction between bees and microbes. And this is just from a simple Web of Science search that I just ran the other day, looking at papers that use the phrase bee microbiome over time. And as you can see here, there's been a sharp increase in the number of papers in this field in the last decade or so. But this field is not new by any means. And in fact, it's been people have studied these interactions for quite a long time, particularly in the role that microbes play as bee pathogens. And this has been particularly well studied for species of economic interest like the European honeybee Apis mellifera, for which we know a lot about the bacteria, fungi, and viruses that infect and harm these bees. So this has been kind of traditionally where these bee microbe interaction studies have started. But more recently, the benefits of these associations have been recognized. So in the past decade or so, it's been realized that social bees actually host a core gut bacterial microbiome that is really conserved and passed between generations. It's composed of just a few bacterial taxa shown in this figure on the left. And these bacterial taxa provide numerous benefits to their host. These include improved immune functioning of the bee and protection against pathogens, as well as improved nutrition through digestion of food resources. So we know there's been a shift in how we view these interactions, and there's been more appreciation of the positive role that these microbes can play in bee health. However, the vast majority of these studies have really focused on bacteria that interact with bees. But the bee microbiome is really complex and diverse and includes diverse microbes. And if you hadn't guessed from the title of my talk today, I'm particularly interested in the fungi that are associated with bees. And these fungi are pretty commonly found associated with bees. They can become really abundant in bee hives and bee bodies. And we actually have quite a bit of research that exists on these interactions that goes back 5 decades or more. So again, while most early studies on these interactions really focused on the pathogenic fungi, there's also was some early pioneering work by Martha Gilliam, who characterized other fungal members of the bee community in the 70s. So there is a lot of research out there, but it's really hard to compare between these studies over time because there have been really vast changes in the methodology that we use to characterize these communities. A lot of this early work was more based on culturing of fungi, and more recently, sequencing technology has become more accessible, and there's been a lot of movement toward that. And in addition, fungal naming conventions have also changed pretty drastically over time, and this has made just comparisons between studies and synthesis of knowledge in this field very difficult. So this was why we decided to try to write a review paper to synthesize what we know currently about bee-fungi interactions and to figure out where our gaps in knowledge are, what we don't know yet. So with that in mind, we wrote this review with 3 main goals. First, to just identify some common non-pathogenic fungi associated with bees. Second, to determine how these fungal communities differ across different bee-associated habitats. And third, to summarize what we know about what these fungi are doing, their roles in bee health and behavior. So before I get into the results, I just want to talk a little bit about how we actually characterized these fungal communities. So we reviewed about 148 papers on these interactions, published from 1972 to 2023, and this covered 81 bee species. We considered fungal communities in 3 bee-associated habitats, these being flowers, the nectar and pollen, bee nests, stored food, as well as the bee bodies, both externally and within the gut. And for each of these papers, we ranked the top 3 most abundant fungal genera that we found associated with each bee species. And we decided to use this ranking approach because, as I mentioned before, differences in methodology make comparison using any kind of quantified method kind of difficult. So we decided to rank the genera instead, and for a culture-based study, this was often based on fungal colony counts or frequency of detection, whereas for sequencing-based detection, this was often based on read number or relative abundance. And then we compared communities across habitats and bee species using these ranked fungi. So to get into our first result here, to identify these fungi associated with bees, I'm going to be showing you a heat map, but I just wanted to take a second to walk through what are on the axes here. So on the y-axis, we have our top 14 fungal genera that we found associated with these bees. And then on the x-axis, we have all of the bee species represented by the different studies colored by bee tribe. And I wanted to point out a little bit more about these bees that we have represented here. These first three groups on the left are all of our social bees. So these are bees that are probably most familiar to us, bees like honeybees, bumblebees, and stingless bees that live in these large colony structures with many, many conspecifics. However, when we look at the general makeup of bee species totally, we see that of 20,000 total bee species, about 77% of them are actually solitary. So they don't live in colonies, and instead one female is provisioning her offspring alone and collecting floral resources on her own. And our solitary bees are represented on the right side of this bee phylogeny, shown mostly in carpenter bees, mason bees, and leafcutter bees. So you can see that we definitely have many more social bee species represented by these studies than solitary bees. And I'll get into that more later, but I just wanted to point that out before we interpret this data. So for each bee species in a column, I'm going to be showing this heat map that shows the presence or absence of each fungal genus within the top 3 genera for that given bee species. So if we look at this data across all of our bees, we see that there is a lot of variation in the fungal communities of these bees. There isn't one particular genus that is present across every single bee species that these studies covered. However, we do see some interesting patterns start to emerge. In particular, we see that yeasts, represented at the bottom of this fungal phylogeny here, are pretty commonly associated with bees in general, much more than the filamentous fungi that are at the top of that phylogeny. And within the yeast, we see these 2 groups in particular are really common. Zygosaccharomyces and Starmorella both are really often associated with bees, and particularly social bees. So we find that these yeasts are present across bee species, these Starmorella and Zygosaccharomyces yeasts. So then we were also curious to understand how these fungal communities differed across different habitats. And as a reminder for this, we looked at flowers, nest provisions, and the bees themselves. And I just wanted to talk a little bit about what these nest provisions actually look like. So nest provisions are just modified floral nectar and pollen. Bees collect this food at flowers, bring it back to the hive, and then modify it in some way for storage. So examples of this in honeybees are honeybee honey, which is just concentrated nectar that has been dehydrated, resulting in a very sugary solution. Whereas honeybee bee bread is much more of a solid provision that is made mostly of pollen mixed with some nectar to create a mostly solid mass. However, again, honey is only produced by social bee species, and solitary bee provisions often look pretty different. They look more like the bee bread of honeybees, in that they are mostly semi-solid masses of nectar and pollen. And you can see the egg laid on this mass. And again, this differs by solitary bee species. Some of these provisions are much more liquid than others, but I just wanted to point out kind of how the variation in these provisions might also affect the fungal communities that we see there. So to go into our results here, we have our habitat on the x-axis, and then the occurrence of each of our top fungal genera on the y-axis. And if we just look broadly, we see that fungal communities across these habitats are pretty similar. They have a lot of the same taxa, but just in different proportions. Flowers definitely stand out as being the most distinct. They contain mostly the nectar specialist yeast Mechnicovia, as well as Oreobasidium and Starmorella. Whereas when we look at bees and their stored provisions, we see that their communities are really similar and are mostly dominated by Starmorella and Zygosaccharomyces, which makes sense considering what we saw in the heat map earlier. So in this case, bee guts really do seem to reflect mostly pretty much whatever they're eating. We don't see specialized communities that are really consistent like we do for gut bacterial communities of bees. However, we do see a pretty big difference between flowers and stored provisions, which is interesting because, as I mentioned before, nest provisions are just modified floral provisions. So why do we see such a big difference? And I've kind of gotten into this a little bit before on the previous slides, but bees are modifying these provisions in ways that make them a little bit more hostile to microbial growth in general. As I mentioned before, they really increase the sugar content to very high levels. Honey can have well over 70% sugar in it. And in addition, as the sugar content increases, the pH of these provisions also decrease. And this is further decreased by the proliferation of lactic acid bacteria in these provisions. So the result is a very, very high sugar content, very acidic environment. And these kind of conditions are hard for many microbes to grow in. So this leads to a specialized yeast community that can tolerate these conditions, as shown in the proliferation of Starmorella and Zygosaccharomyces. Great. So we've talked about how these fungal communities differ across habitats and just what these fungal communities look like in general. But what are these fungi actually doing? What are their effects on bee behavior and bee health? And this result is much less well represented by the literature than the first two. A lot of people have characterized fungal communities, but there have been many fewer studies on the actual function of these communities. However, we do see some interesting patterns start to emerge. In particular, nectar yeasts, when they are present in the nectar of flowers, seem to modify bee behavior in pretty predictable ways. Bumblebees in particular seem really attracted to the yeast Mechnicovia when it's in the nectar of flowers, as shown by this 2019 study, where they either inoculated a plant with nectar yeasts or left it microbe-free as a control, and then they measured pollinator visitation rate and seed set of the plant. And they found that not only did the yeast being present in nectar increase the visitation rate of pollinators, but this also led to higher seed set. So it seems like this is beneficial for the plant to host the yeasts in this case. And it's also been hypothesized that these yeasts are beneficial to foraging bees because they might provide an honest cue to bees that signifies that there is nectar present, because as the yeasts are growing and metabolizing that nectar, they're producing volatile cues that bees can use to forage. There have also been some really interesting studies of these bee yeast interactions in stingless bees, in particular this stingless bee Scaptotrigona that relies on the yeast Zygosaccharomyces for its development. So this is an example, a figure showing the brood cell of this bee species, and you can see in panel C here, there's a lot of white filamentous growth along the side of the brood cell, and that is the yeast Zygosaccharomyces growing in this very liquid provision. But as you'll see in panels D, E, and F, that white growth disappears. And this is because this bee is actually eating this fungus. So here's a video of this bee kind of circling around its brood cell. It's eating the provision, but it's also eating that fungus off of the wall of the brood cell. And it turns out that this fungus is almost obligately necessary for these bees to survive. So they tested survival of these bee larvae with that fungus present in the light gray bars, or with that fungus removed via sterilization, shown in the black bars. And you can see just visually that survival drops pretty steeply from around 76% on average to only 8%. So in this case, this fungus is very, very important for bee development. And this is because these yeasts are providing sterols that are necessary for the bee to pupate. And without these sterols, it cannot progress through development and they die. So overall, we see that we do have some common yeasts associated with bee species, namely those genera Starmorella and Zygosaccharomyces. We see that the conditions in the bee nest lead to a distinct specialized fungal community that could be found there. And we also see that these fungi are impacting bee health in numerous ways, as well as attracting bees to flowers and affecting their foraging behavior. However, our knowledge of these relationships really remains incomplete and is pretty biased based on what information we have available to us at the moment. One big bias is that most studies on this topic focus on social bees and in particular honeybees, shown in this red bar here. Honeybees were represented by 54% of the studies that we looked at. And if you'll remember this figure I showed earlier, this is not representative of bees more broadly, as 77% of them are actually solitary. And if we color in the other social bee groups also with these red bars, we see that the remaining gray bars are actually our solitary bee species that have been studied. So it's very, very few studies on these species. There's a lot more to learn about these associations. In addition, we also see that very few studies focus on larvae and most actually focus on adult bee stages. And I think this is a really promising open area of research, especially given the importance that we know that these fungi can play in larval health. And finally, studies on function are lacking in general, as I mentioned earlier, particularly how these yeasts are impacting bee nutrition and defense against pathogens are very open questions right now. However, I say this, but in the relatively short period of time since this review has been published, we've already made a lot of advances in this field. And I want to talk about one really cool example today by my lab mate, Sean Christensen, who works on the solitary bee Anthophora bomboides. This bee lives on the California coast. It nests in cliffs that face the ocean. This is what one of its brood cells looks like dug up. And this bee actually has surprisingly consistent associations with fungal symbionts. So Sean looked at the microbiome of these bees across development, starting from egg all the way through pupation. And here you can see the abundance of different fungal genera are represented by different colors. And you can see there's a lot of this blue bar throughout the development of this larvae. And that blue bar is actually the genus Monoliella, a yeast-like fungus that is consistently associated with these bees across sites and across years. So it's a very, very consistent association. What the fungus is actually doing is still not quite known, but it seems like it might be playing an important role in development. And it definitely shows that there are a lot of promising avenues of research with these solitary bees. So while there's a lot left to learn, we're learning a lot of new stuff every day. And I, for one, am very excited to see what the future of this field looks like. So with that, I'm just gonna hang on my reference slide for a little bit here in case people wanna look into these later. And then I'd like to thank everybody for listening. Thank you again to Max and FEMS for inviting me. And I'd be happy to take any questions.
Thank you so much. And again, please put in the questions in the Q&A, and we're going to get back to them at the very end of the session. And of course, if you want to read more, there's the wonderful mini review, Bees Just Want to Have Fungi, to look in, and you've seen the links on there. So thank you very much for that introduction to bee microbiology. And we're going to continue with bees. And Suni Matthew from University of Turku is examining the role of herbicides and thinking about what we just heard on how important the food is. Of course, using herbicides that kill the plants that are maybe important for feeding the bees. It's going to be interesting to see what glyphosate is doing. So Suni, to you.
Thank you, Max. Yeah, thank you, Max, for the introduction. And again, thank you everyone for joining us on this webinar today. So my topic is glyphosate and a glyphosate-based herbicide affect bumblebee gut microbiota. Glyphosate-based herbicides are the most widely used herbicides around the world, and it is a broad-spectrum weed killer. It is used in agriculture, horticulture, silviculture, in urban gardens and lawns. and it's used for various other applications apart from weed killing as well. It is used for crop desiccation during pre-harvest, for renewal of temporary grasslands, and is also used for termination of cover crops. With the development of glyphosate-resistant crops, the use of glyphosate-based herbicides have increased immensely around the globe. The mechanism of the active ingredient, glyphosate, to kill the weeds is by blocking the shikimate pathway in plants. And here you can see a diagrammatic representation of the shikimate pathway. So here you see that the glyphosate can bind to the protein or the enzyme EPSP synthase, and then it blocks the production of the compound 5-enol pyruvyl shikimate 3-phosphate compound. So this then again blocks the synthesis of the 3 essential amino acids, tryptophan, phenylalanine, and tyrosine. Now, once the synthesis of these amino acids are blocked, the plant is unable to produce compounds like phytohormones, such as indole acetic acid, or then secondary metabolites, which then interferes with the survival, growth, and defense mechanisms of the plants. So this is how the glyphosate-based herbicides eventually kill the weeds off. Now, the manufacturers of glyphosate-based herbicides claim that they are safe for the environment, mainly because of 2 reasons. First, they are claimed to be quickly degrading in the soil within 2 weeks of application. But several studies have shown that this is not the case. Glyphosate residues can remain in the soil for at least 6 months and can range up to several decades, depending upon the soil texture, soil composition, and also the climate of the geographical location where these herbicides are applied. Again, the second reason why the manufacturers claim that glyphosate-based herbicides are safe for use is that the shikimate pathway is absent in animals, so then it's considered safe for use for animals or humans. But this has also been contradictory because results have shown that they affect the health of different animals. And then the factor that is mostly concerning is that several bacteria and fungal taxa have this shikimate pathway. So then application of glyphosate-based herbicides, how does that affect the microbes that have the shikimate pathway that are present in the soil, or then those microbes that are associated with plants or animals? Now, the Helander group at the University of Turku, of which I'm part of, has been studying this research problem. As I said, glyphosate remains in the soil for several decades, especially in the northern ecosystems where the cold climate prevails. And our studies have shown that the glyphosate residues in soil affect microbial community composition in non-target crop plants. It can also affect microbial community composition of insects such as Colorado potato beetle. And then it has shown to cause adverse effects in embryonic development and oxidative status of Japanese quails. We have also studied the effect of glyphosate-based herbicides on plant performance, such as plant germination, the growth of plants, and hormonal homeostasis. And they showed, again, adverse effects. And finally, we have also shown that herbicide residues in soil can affect plant resistance and plant defense mechanism, either by changing the quality of the plant or by decreasing microbe-mediated plant protection mechanisms. So from these research findings, the question led to how does glyphosate-based herbicide exposure affect pollinators? And as our first speaker, Daniel, had presented a really nice data about how the effects of pesticide use on pollinator decline. So as she said, about 75 to 95% of the flowering plants are pollinated, and that includes more than 180,000 flowering plants and more than 1,200 crops. So we now have these two studies where we studied the effect of glyphosate-based herbicide on bumblebee visual recognition and on their cognitive functions, and it was found that they affect their fine color discrimination and learning and memory of bumblebees. and our study species is Bombus terrestris. Now, this led to the research question of this article that we published in FEMS Microbiology Ecology, that how does glyphosate or glyphosate-based herbicide modulate the composition of bacterial communities in bumblebee gut? So I can say a bit about the experimental setup in our study. As you can see here, these are two wooden chambers where the beehives were located. And this is connected by an acrylic tunnel where the bumblebees can then go to the foraging arena where the bees are fed, either with control that was 60% sucrose solution, or then either of these two treatments. It was either pure glyphosate or then glyphosate-based herbicide. And here in this study, we used the product Roundup Flex. So this was given at two concentrations, at a low and high concentration, over a period of 5 days. And the bumblebees were collected at a 3-day time point and a 5-day time point. So once the bumblebees were sampled, the gut was isolated and then DNA was extracted, followed by 16S rRNA gene PCR, and then high throughput sequencing by Illumina MiSeq platform, and then the data was analyzed. Now, discussing about the results, as you can see here, this shows the taxonomic distribution of the bacterial communities. And here you see there are bacterial community composition of the 5 bees collected for the two treatments, glyphosate or Roundup. And then here you see that this is the community composition of the bumblebees collected as controls, or then of the bumblebees collected at 3-day time point or 5-day time point. Now, one of the key points here is that you can see about 11 dominant genera, and this shows that the bumblebee gut bacterial community has a low diversity. Again, looking at the alpha diversities, we see here that the glyphosate treatment increased the bacterial diversity at low concentration, but then Roundup decreased the bacterial diversity at low concentrations. Again, glyphosate increased the bacterial diversity over a period of 5 days, but then here for Roundup, we don't see any significant observations. So here we see that the bacterial community composition was significantly shifted by both glyphosate and glyphosate-based herbicide treatments. And the most dominant genera that brought about these differences included Klebsiella, Bacillus, and Snodgrassella. So with the relative abundance, we see that there was an increase in the relative abundance of Klebsiella and Bacillus, but then there was a decrease in the relative abundance of Snodgrassella algae. Now, this is a bit concerning because Snodgrassella has been found to be beneficial for the bumblebees as it is involved in pathogen regulation. So if there is a decrease in the relative abundance of Snodgrassella, how does this affect then the health of the bees? Again, the third key result from our study was from the bioinformatics tool that has been developed by a collaborator, Pere Puchbo. So with this tool, we were able to predict the glyphosate sensitivity of the bacterial taxa present in bumblebee gut. Now we know that the EPSPS protein sequence as present in the bacteria has conserved amino acids, and it is with these conserved amino acid structure that we determine whether the EPSPS protein sequence is class 1, that is glyphosate sensitive, or it can be classified as class 2 glyphosate resistant. So if you recall that glyphosate inhibits the EPSPS protein. So with this information, we were able to identify whether the bacteria is glyphosate sensitive or resistant. Now, the EPSPS protein sequence is phylogenetically conserved based on the information from the database. So with this, we found that more than 80% of the bacterial taxa present in the bumblebee gut is glyphosate sensitive. So then the question arises, if the bumblebees are exposed to glyphosate-based herbicides, how does this then affect the abundance and diversity of glyphosate-sensitive bacterial taxa? Summarizing the results from our study, glyphosate and glyphosate-based herbicide treatments did significantly shift bacterial diversity and community composition in bumblebee gut, and changed the relative abundance of functionally relevant bacterial taxa, for example, Snodgrassella, and then more than 80% of the bacterial taxa in bumblebee gut are predicted to be glyphosate sensitive. So our future perspectives is to study, as you can recall, glyphosate and glyphosate-based herbicides gave different trends on the bacterial diversity measurements, and this can be due to the co-formulants present in the herbicides. Now, it's obligatory for the manufacturers to give out information only about the active ingredient glyphosate, but then the information about co-formulants is proprietary, and it's not mandatory to give out this information on their marketing labels either. So we do not know what kind of co-formulants or concentrations of co-formulants are used, and it can also vary geographically. So the effects of co-formulants should equally be studied. That is, the effect of co-formulants on bumblebees or non-target organisms should be equally studied as in glyphosate. Again, from our study, we see that it affects the bumblebee gut bacteria. So then we know that they are beneficial for the bees. How does this affect the cognitive functions and health of the bees? And at a higher trophic interaction level, how does this affect pollination and ecosystem functioning? And in our lab, we are presently trying to identify the bacteria from the bumblebee gut and link their glyphosate sensitivity to cognitive functions and health of the bees. So with this, I conclude my topic and thank everyone for listening to me. And this is the website link for our group. So if you want to know more about our research, so feel free to click on this link. Thank you very much.
Thank you, Suni. And I see there's already a bunch of questions coming from you, but we will wait for them till the end. I also have a couple of things I want to ask you, but thank you very much.
Thank you.
We will go now from bees to beetles and look at another group of insects. So Teresa Veselska from the Czech Academy of Sciences. And again, there's been, of course, a lot of concern with beetles and other insect infestations in the forest. So looking forward to hearing what you have.
Thank you, Max, for invitation to this wonderful webinar. And so let me share. Thank you. So hello, everyone. My name is Teresa Veselska, and I am from the Institute of Microbiology of the Czech Academy of Sciences. And my presentation will go around our paper published in FEMS, entitled Gut Core Microbiome Changes Throughout the Life Cycle and Season of the Bark Beetles, Beetle Ips typographus. However, in my presentation, I would like to go beyond this paper and present all our projects. So I would like to start with acknowledgement of all people working on it, and I would like to say my thanks to our researcher at our institute, especially to Miroslav, Karel, Martin, and Barbara, and then Tian from Helmholtz Center for Infection Research in Germany, and Paula Zaki and Ezekiel from University of Salamanca in Spain. So let me introduce bark beetles. They are extremely diverse group of insects. They have around 6,000 species around, and they have various ecologies and feeding habits. The most common is that they bore under the bark of a tree and feed on phloem tissue, and they are mostly saprotrophs. It means that they feed on dead trees. However, small proportion of them could also kill healthy trees. So that's why bark beetles have a huge global social economical impact. These tree-killing bark beetles belong to different groups. And the first is called aggressive bark beetles. And these beetles bore into a healthy tree and cause its diebacks. And the second is secondary bark beetles, and these beetles usually feed on dying or freshly dead trees. However, if some large part of forest is destroyed, for example, by a storm or weakened by some stressful conditions, for example, long-term drought, they can increase their population size, and then under massive attack, they can kill a large part of healthy trees. So both of these groups could destroy a large part of our forests. So that's why they are so important. However, bark beetles have to cope with a very harsh environment because the phloem tissue is very hard to degrade. And there is a very limited amount of key nutrients like nitrogen, phosphorus, sterile vitamins. And phloem is also full of toxic plant secondary metabolites, which really can kill bark beetles. And this environment is also highly competitive. So that's why they are often associated with filamentous fungi, which can degrade this plant cell wall. And as they grow distantly into plant tissue, they accumulate in their hyphae these essential nutrients. And then the beetle can graze on this hyphae in the phloem tissue and acquire these nutrients from hyphae. These fungi also can degrade toxic plant secondary metabolites and can produce biologically active compounds like antibiotics. So they can protect beetle against enemies. And they are also called blue stain fungi. It's because as they grow inside plant tissue, they colorize it in blue. So you can see it on the picture. And so they are very conspicuous and long-term studied. However, very little, however, it's not only fungi that can help, but also beetle, because this beetle carry fungus under the bark of a tree into a phloem tissue, which is otherwise hardly accessible for the fungus. And so beetle provide fungus a new source of nutrients. So both organisms are happy together, and so the symbiosis is called mutualism. However, besides this filamentous fungi, very little is known about all other microbes. So we pose this question and try to answer them during our research. So the first is what is the diversity of microbial community associated with bark beetles, how it changes throughout the life cycle of the beetles, and how it changes between seasons, and what are the functions of microbial community. And we studied on Ips typographus, which is Phloemophago secondary bark beetles. And this beetle is the most significant pest of European spruce forest. This is the typical gallery system which you can find under the bark of a tree. And this beetle can destroy very large part of our forest because our spruce forests in Europe are planted outside a native region, so they are weakened by long-term drought and so on. So it causes large-scale damages. This beetle has usually 2 generations per year, one in the spring and second in the summer. But because of global warming, it could have up to 3 generations. So the pressure of that beetle to our forest intensify and will be even worse in the future. The life cycle of Ipstifographus begins when males bore under the bark of a tree into phloem and attract females. And these females then create corridors inside phloem tissue and lay eggs there. And these eggs then hatch into larvae, which then feed on phloem tissue, and which is colonized by fungus. And after some time, they create pupal chamber, and in this pupal chamber is beetle transport into young beetle, which then leave the spruce tree and search for a new one. And the whole life cycle takes around 10 weeks to complete. And you can see then that almost whole life cycle is hidden under the bark of a tree. So what is known about microbial symbionts of Ipstifographus? So it's known that it transfer on its body surface these ophiostomatal filamentous fungi, these blue stain fungi, Endoconidia fora polonica, Ophiostoma bicolor, and Grospania penicillata. All of them are known to be able to degrade toxic secondary metabolites of plant. So they are really important for the beetle. And you can see in this picture how they looks like in a pupal chamber of Ipstifographus. However, very little is known about the rest of microbial community. We studied, we sampled freshly infested spruce tree in a niche bore forest near Prague and transferred logs into our institute. And then we sampled the whole life cycle of the beetle, and we did it in spring and summer generation. And we studied a microbial community of gut of this beetle by DNA metabarcoding, and also we assess functions of its microbiome by metatranscriptome analysis. So here are our results. The first, from this plot, you can see that both bacterial and fungal microbiome differs between seasons. But if we look closer to its microbiome composition, you can see that the most abundant species of fungi and bacteria are very similar in the spring and summer. However, they differ in their relative proportions. So it could mean that these species have different requirements for their growth. Some need, for example, lower temperature or higher moisture. So this may be the reason. And if you look more closer to fungal microbiome, we found very interesting results that these well-known filamentous fungi associated with bark beetles took less than 20% reads of total reads. So they are present, but not the most abundant. And the most abundant were yeast. They took more than 80%. So it's very similar to bee situation about which Daniel spoke about. And if we look at the RNA metatranscriptome analysis, we again see that yeast are very active there. And it means that they are not only present, but also active. So it means that yeast are really important for Ipstifographus. And it's even more clearly seen when we look at the core fungal microbiome. So now I need to say that we define core microbiome as a species that are present throughout the whole life cycle and in both generations of the beetle. So they create some stable and persistent community. And 9 of 10 taxa belonging to core fungal microbiome are yeast. So again, yeast are probably very important. When we look at bacterial microbiome, we can see that Enterobacteriales, and especially genus Erwinia, is the most abundant. And again, when we look at metatranscriptome analysis, Enterobacteriales are very active. So it means that this order is really important for the beetle. And when we look at core bacterial microbiome, we identified 12 different taxa, and the most abundant were Erwinia typography, Erwinia serenensis, Pseudomonas bohemica, and Pseudoxanthomonas spadix. Then we can move to our functional analysis. We made several methods we used, and first of them is genome sequencing. Then we run in vitro enzymatic test, antibiotic production, and metatranscriptome analysis. And here I would like to show our result from genome sequences of the most abundant yeasts. And first we look at their ability to degrade plant cell wall. So we found, you can see it on the left picture, we found enzyme active on cellulose, hemicellulose, and pectin. And then we wanted to know whether they are really cleaving these substrates. So we run in vitro enzymatic test and measure enzyme activities. So when we look at these plots, you can see that none of the yeasts are able to cleave these complex molecules like inside the chain. So it means that they are not really good at cleaving plant cell wall, and they rather use some simple sugar for growth. But the yeasts are really important for beetle nutrition because they can synthesize vitamin B6, which is essential, and also all amino acids. Then we could look at the functional role of bacteria. These are the results of our colleague from Spain, and they run very nice in vitro analysis. And they found out that most of bacteria associated with Ips typographus are able to degrade cellulose, pectin, and starch. So they are really important for plant cell wall degradation and provide carbon to beetle. And they are also very good at fungal inhibition. Yeah, so they protect beetle against some entomopathogenous fungi. So they protect the beetle, and they do it also via siderophore production. And here I would like to share with you our results from metatranscriptome analysis. These are just preliminary results. We still are working on that. So these data are not published, not yet. But I would like to show you that these results are very nicely confirm our previous results from genome analysis and in vitro tests, because we found out that fungi and bacteria all are able to synthesize all essential amino acids, vitamins, and cofactors. And especially bacteria are active on plant cell wall degradation. So to sum up, I would like to say that microbiome of Ips typographus changes between seasons. Ips typographus has a core microbial community. Yeast and bacteria, Enterobacteriales, are particularly abundant, and microbial community is involved in providing of essential nutrients and degradation and detoxification of plant tissues. So thank you for your attention.
Thank you so much. And yeah, there's already a bunch of questions, and I appreciated your work on looking at the activities and how that's affecting it. And actually, while I'm looking through the other questions coming in there, the seasonal difference that you're observing, do you know what is the driver on that? I mean, the seasonal effects, of course, in soils have been seen, but would you know what would be driving this change?
Yeah, probably some abiotic factors. Yeah, it's known from these filamentous fungi that, for example, because it's probable that these microbes are very similar functions, that they do the same thing. Yeah, we saw it very nicely when we analyzed genome of yeast, that they have very similar very similar functions. But they probably have different requirements for growth, some temperature, moisture, something like that, that could drive it. So probably this is something that is really important. And yeah, it's known also for another filamentous fungi associated with the Dendroctonus bark beetle in US. And also is the same thing that in some area which is, yeah, this is probably because of abiotic factor. One of these fungus are like more abundant, and the another area is less abundant, and the other is more abundant. So probably abiotic factors are important.
Yes, but you're looking here also at relative abundance. Do you know about absolute changes? I mean, could it be that in the spring you still have low numbers in general, and then there's much more growth, and it's selective growth of different members?
No, that's not like that. No. It's more than the spring is like more diverse. It's probably because Ipstipographus can overwinter under the bark, or also like in the leaf litter on the ground. So they acquire some other microbes from these environments and take them with themselves to new tree. So the spring season is much more diverse than summer season. So then there's a shift, yeah, which maybe is driven by the consumption of phloem tissue.
Okay, thanks. There are a couple of questions here for Suni and Danielle, and I'm just combining a couple of these in there. And this has to do, of course, between this aspect of detecting an organism versus is it truly active. And so would you know, in terms of the bee gut microbiota, which members of the fungi or bacteria are active versus just maybe resident or transient?
Yeah, so it's a really good question, and I think it's one of the downsides of just like pure sequencing approaches, is that you are never quite certain if that is from viable cells or if it's from, as has been mentioned, like transient members of the microbiome. I think that at least for the bacteria, there has been some like transcriptomic work that shows that they truly have functions in there, and I'm sure that Suni knows more about that than I do. But for fungi in particular, that kind of research is not as common, but from what we do know, it certainly seems that at least within the bee gut, they're not playing a huge metabolic role from the transcriptomic data that we do have. So at least for the bee gut, it seems like they are mostly transient through there and mostly from the food.
Okay.
Yeah, I can just add to that based on the studies that we do, like based on glyphosate sensitivity. For example, this is for the bumblebees, this is a nice study species when you look at the perspective of scientific research, that there is low diversity of bacterial community. And then if we are able to pinpoint on their EPSPS protein, for example, the transcriptional activity of EPSPS protein during these treatments, then that might also show that how active these bees are, or the bacteria in the bee gut are. Yeah.
So I have also a follow-up question regarding your experiment with glyphosate versus Roundup. And of course, there you have all the other additives that are part of the formulations. And I know that there's been a lot of discussion also on their roles and so on. Any idea of the difference? I mean, I looked at quickly from some of your data, it seems like the community structures were different depending on whether you had a glyphosate treatment or the Roundup treatment. And so what roles could these various surfactants and additives have?
Again, these are chemicals, and for example, you see here that the glyphosate does have an effect on changing the bacterial community composition. So then the co-formulants are adjuvants, or then there are substances that help in penetrating the glyphosate more into the plants. Those are the co-formulants that are used with glyphosate. So then and you see that there is a difference in the trend of changing the diversity if it's glyphosate or glyphosate-based herbicide. So that could, we would think that it comes from the effect of the co-formulants.
Okay, yeah. So there's a lot of combinations of that to look at. And actually, this was one question here from the Q&A about the concentrations that you used in your experiments versus field conditions.
Mm-hmm. Yeah. So the low concentrations were comparable to other studies where they have tested the glyphosate on bumblebees and honeybees. And then in field conditions, about 13% to 26% is used in field conditions. And they have shown studies that maybe like the concentration that we used, the high concentration that we used in our study can still be found, those glyphosate concentrations can still be found on the field plants. So then the bumblebees, of course, then it gets mixed with the nectar and pollen, and then the bumblebees can get exposed to these high concentrations. So that's why we selected this kind of concentration for the study.
Okay, here's one that just came in to link all of this into climate change. And I can actually see a couple of different aspects, both for Teresa and Danielle on this. This is to Danielle. In terms of background, and is it possible to do a correlation between the data on climate change and bee fungal interactions? Are there things that are changing for the bees with also maybe changes in plant cover, but also temperatures and so on?
Yeah, it's possible. It'd be hard to do with the data that we currently have, just because a lot of the older studies use different methods than the newer studies, which makes like comparison across time with climate data kind of difficult, since detection method affects community so much. But I do think that it's possible that there would be an effect more generally. Certainly, even when assembling communities within the beehive, we see that temperature has a really big effect, where if hives are kept at their normal temperature, a very different community establishes than if they're warmed a little or cooled a little. So beehives are really sensitive to those kinds of changes. And flowers as well are also pretty sensitive to temperature and what kind of community can establish there. So I wouldn't be surprised if there was an effect of climate change and warming on these communities, but I don't think that we have that data yet.
So what about the bark beetle infestations? Of course, a lot has been linked to the poorer health of the trees, but are there other things that you can see on microbiome interactions?
Yeah, so I think that is very similar, as Danielle said, that we can, it's hard to say when we don't have the data, but of course we can see that, for example, bark beetles that bore into the same tree, but they have different preferences. For example, some are on trunks and others on twigs. They have really different microbial communities. So really abiotic conditions like this moisture, temperature, and these things really shape the microbial community. So yeah, I think that it will change with global warming and so on, but hard to say without data. Yeah.
And also in the seasonal changes in terms of activity of the microbiomes. This was a question here from the Q&A.
Yeah. So at least in our data, we found that even that these microbes change. Yeah, they have different proportions. The functions is the same. So they like they have the same functions and they just change their proportions because of these abiotic factors. But I don't know if it's changed too much what's happened, but at least at these days, we can say that the functions are the same.
Okay. And anything about the lignocellulosic hydrolysis? So of course, you saw a lot of species with utilization of sugars and so on, but a lot of it is, how are you seeing degradation of cellulose and how that would play a role in all of this?
Sorry, you mean how this global warming have an effect on cellulose degradation, or what?
No, this is more about the microbiome activities in general. I mean, what's their role in degradation of the lignocellulosic material, and is that actually feeding the bark beetles as well?
Yeah, so definitely bacteria can do it. They have full pathways. Fungi, we can for yeast, they probably don't do it. Some filamentous fungi can do it. For example, these Ophiostoma bicolor have full pathways, so they, I think that they collaborate to each other and use the metabolic, it's called somehow like external gut, that they combine their abilities together, and together they have this full function for this ecology. Yeah, so at least the bacteria and filamentous fungi can do it. Yeah.
Okay, and I think we're going to take one more question here, and this is there a relationship between the enormous decline in honeybee populations and their microbial biomes, their diversity or composition? So again, is there, and again, it could of course just be links. I'm thinking of, are there new pathogens that are coming in, or other aspects, or are you losing beneficial microbes? So either Suni, Danielle, both of you, yeah.
Yeah, so there has been study that, for example, in bumblebees, where they showed that there was a decrease in lactobacillus taxa, and then that was connected in affecting the learning and memory of bumblebees. Again, we have also seen in our studies that, like I showed, there was connections when there were treatments with glyphosate that it impaired their learning and memory. So then there are difficulties for bumblebees to find back their way to their hives. So all these behavioral changes could affect, again, the health of the bees, or then let's say there is a relative abundance increase in or decrease in beneficial fungi that helps in pathogen regulation, as we see here in Snodgrassella. So they can be affected easily by pathogens. All these results show that they can be a major cause for pollinator decline.
Danielle, anything to add?
No, that sounded great, Suni. I think you covered it.
Okay, there are a couple of very technical questions in the Q&A. I'm going to actually, just to you who asked them, to direct you to the papers and directly to the authors to check with them. I think that's going to be more beneficial than trying to cover it up here over the webinar. So again, there are some other questions, or if you have additional ones, then please contact Danielle, Sunia, or Teresa directly. And with that, actually, I want to thank the three of you for wonderful talks, wonderful papers, and looking forward to hearing more of what you're doing. And thank you, everyone in the audience from what I saw all over the world joining in on this webinar again. And keep in touch. We'll take a look at what topics we'll continue on in the fall. And so if you have any suggestions, please send me a note as well. And so thank you also to Joe and everybody else who's been making this work so well. So thank you.
Thank you very much for organizing this.
Yes, thank you so much.
Yeah, thank you very much.
Okay, bye everyone.